Bismillahirrahmanirrahim. Assalamu alaikum students. Hope you all will be fine and safe. Welcome you all in lecture 3 of biology. In this lecture, we will discuss the factors affecting the rate of enzyme action and mechanism of enzyme action in detail. Before starting our lecture, a short recap of the previous one. In the previous lecture, we have discussed the characteristics of enzymes and uses of enzymes in different industries in detail. Our today's topic is very important and very interesting, that is on factors affecting the rate of enzyme action. Every enzyme, it works in its specific environments, and each enzyme has different factors that can affect its activity. What these factors will do, they may change the chemistry of the enzyme or they may change the shape of the enzyme, and in both the cases the enzymatic activity, it will harm or it will affect. There are different factors that can affect the rate of enzyme action. They may be inhibitors, they may be cofactors, they may be enzyme concentration, temperature, substrate concentration, and pH. But most important factors that affect the rate of enzyme action are temperature, substrate concentration, and pH. Now, the first factor, that is temperature. Temperature is very important for enzyme catalyzed reactions. Why it is important? Because every enzyme, it works at its maximum rate at a specific temperature, and that temperature is called optimum temperature for that enzyme. You can easily understand it by the example. In human body, different enzymes are working. Some are involved in the process of digestion, some are involved in the cellular respiration, some are involved in excretion. So all these enzymes, they work at a specific temperature, and our bodies, means humans body's internal temperature is 37 degrees centigrade. So 37 degrees centigrade is the optimum temperature for human enzymes, not for the others. Similarly, the birds, they have high body temperatures than mammals, so their enzymes, they work at their optimum temperatures. Plants, they have their own temperatures. Other fishes, aquatic animals, they have their own temperatures. So similarly, all the enzymes, they have their own specific optimum temperatures at which they work. So what the temperature will do? What's the effect of the temperature on the enzyme? When we provide a temperature to the enzyme, the molecules of the enzymes or the atoms of the enzymes, they start to hit one another. Just like in chemical reactions, you can see, or you can see in case of water, when we give heat to the water, when we heat water, the molecules of the water, they start to collide with one another, and then at 100 degrees centigrade temperature, they start to change into vapors. Similarly, when we rise the temperature, or when we give temperature to the enzymes, their atoms, or their molecules, they also start to collide with one another, and temperature provides kinetic energy to those molecules, and then the enzymes, they give speed to that reaction. But this increase in the temperature, it will be at a specific limit. It has a limit. If we cross that limit, the enzyme molecules, they will hit each other at a very high speed, and then what will happen? The enzymes shape, or it will lost. When the temperature rises to a certain limit, heat adds in the activation energy and also provide kinetic energy for the reaction. But when the reactions are accelerated, when there are more reactions, The temperature, when the temperature is raised well above the optimum temperature, what happens? Heat energy increases the vibration of atoms of enzymes and globular structure of the enzyme is lost. This is called denaturation of enzyme. It results in a rapid decrease in the rate of enzyme action, or it may be blocked completely. You can easily understand the example with the help of the graph. On x-axis there is a temperature and on y-axis there is the rate of enzyme action. You can see at the start when there is low temperature means there is 10 degrees centigrade, the enzymatic activity is slow. But with increase in temperature, the enzymatic activity goes on increasing. At 20 it is more than 10, at 30 it is more than 20, and at 37 degrees centigrade before 40 it is maximum enzymatic activity. This graph shows that the enzyme, it works at maximum speed or at maximum rate at 37 degrees centigrade. If we increases the temperature, rises the temperature further than 37 degrees centigrade or 47 degrees centigrade, then what will happen? There is a loss of activity at temperatures above 50 degrees centigrade. As you can see in the graph, the enzymatic activity is decreasing, the rate of reaction is decreasing, and at 50 or 51 degrees centigrade the denaturation of the enzyme occurs and there is the loss of catalytic activity. You can also understand how the temperature affects the enzymes with this example or with this graph. There is also a temperature on x-axis and percent maximum activity on y-axis. Enzyme activity at 0 degree centigrade is slow, or the enzymes, there is no activity at 0 degree centigrade, but with the rise in temperature you can see there is an increase in the percent maximum activity of an enzyme. The important point is enzyme activity doubles with every increase in 10 degrees centigrade. When we increase the temperature or when we add 10 degrees centigrade temperature, then the rate of enzyme action will become double. Collisions are happening more frequently. The molecules, they start to hit one another, and due to this faster molecular motion, the enzyme shows their maximum activity at their optimum temperature. But what happens at high temperatures? At high temperatures, enzymes, they are denatured. They change their shape. How? What will happen when we rise the temperature to a certain limit? Heat adds on in the activation energy of the enzymes and they provide kinetic energy to the molecules of the enzymes. Reactions are accelerated, reactions speed up. But when the temperature crosses the optimum temperature, heat energy increases the vibration of the atoms of enzymes, their globular structure of the enzyme is lost, and due to this, the denaturation occurs. And when there is a denaturation, there is no enzymatic activity or the enzyme action, it will completely blocked. Our next important factor is pH. What do you mean by the word pH? Potential of hydrogen. pH means potential of hydrogen. All the enzymes, they work at their maximum rate at a narrow range of pH. If there is a very narrow range of pH at which the enzyme works, and that narrow range of pH is called optimum pH. If there is a very slight change in the pH, the enzyme activity, it will block completely. Every enzyme has its own specific optimum pH value. For example, pepsin is an enzyme that digests the proteins. It works in the stomach and and pepsin enzyme, it will become active at low pH, that is acidic pH. And as we know that you all you will learn further in the chapter of digestion that how the pepsin it will become active in acidic pH. Similarly, trypsin is also a protein digesting enzyme, although both are protein digesting enzymes, but they both of them they work at different pH. Trypsin it is working in the small intestine, but it will become active at high pH, that is alkaline pH. So both of these enzymes they are protein digesting enzymes, but they have different pH values. If the trypsin will given the acidic pH, it will not become active. Similarly, if the pepsin is provided the alkalinic pH, it will not become active. So every enzyme, it has its own specific optimum pH value. And if there is a change in the value, the enzymatic activity, it will blocks completely. You can better understand how the pH value it will affects the enzyme action with the help of this graph. You can see the pH value along x-axis and the rate of reaction along y-axis. Enzymes they are most active at optimum pH. In case of, for example, the optimum pH for enzyme is 7, so they will become at their optimum pH, that is 7. Amino acids. Amino acids are the molecules with which the enzymes are made up of. Amino acids with acidic or basic side chains have the proper charge when the pH is optimum. Means if we see the structure of an amino acid, the amino acid has side chains, and these side chains they have specific charge. If there is an optimum pH, then the charge distribution on both the sides of the amino acids will become neutral, and the enzymes they show their specific or maximum activity. If there is a change in the pH value, means the pH value is below the optimum value or high the optimum value, the tertiary structure is disrupted and the enzymes they will not work properly. The third factor that is also most interesting is the substrate concentration. If you recall the previous lecture in which we have discussed how the enzyme they works, enzymes they attach with the substrate molecules, the molecules that act as a raw materials. Those molecules they will attach with the enzymes, then a complex is formed that is called enzyme substrate complex, and that enzyme substrate complex it will, when the substrates they are converted into products and the enzymes they will separate from them. Now what is the role of the enzyme concentration or substrate concentration? Substrate concentration is the amount of, yeah, or the number of molecules in a reaction, a number of molecules of substrate in a reaction. Similarly, if we talk about the enzyme concentration, the enzyme concentration means number of enzyme molecules in a reaction. If enzyme molecules they are available in a reaction, for example, there are 50 enzyme molecules in a reaction, and we are adding 10 molecules of substrate. So the 10 molecules of substrate they will bind with the 10 molecules of the enzyme, and 40 enzymes will left. It means there is more capacity of enzyme, there is more capacity for substrate molecules, so we can add further 10 more molecules in the reaction. Then we can add further more molecules. When if enzyme molecules they are available in a reaction and we are increasing the substrate concentration, what will happen? There is an increase in the rate of reaction. The rate of reaction will speeds up. But what will happen if we kept the enzyme concentration constant. For example, the enzyme concentration will become 50 and we are adding more substrate molecules in that reaction. Then what will happen? A point is reached when any further increase in substrate does not increase the rate of reaction anymore. When the active sites of all the enzymes they are occupied, there are no more enzyme molecules available for the substrates. The substrates they will not find more active sites. So what will happen? This state is called saturation of active sites. Means all the molecules of the enzymes, their active sites will become saturated, and now there is no more enzyme molecules are available for the substrate, and reaction rate does not increase. Here you can see it in the graph. Initially, when if we kept the enzyme concentration constant and we are adding the substrate concentration, this rate of reaction is increasing. But at a point that is marked with a green color, beyond that point there is a straight line. It means there is no change in the rate of reaction. There is no, although we are increasing the substrate concentration, but there is no change on the enzyme reaction. What's the reason? The reason is beyond the green point, enzyme molecules they are available for the substrate molecules, so that's why rate of reaction is increasing. But after that green point, all the enzyme molecules they are occupied by the substrate molecules. We are adding more substrate molecules but no free active sites are available, so that's why the reaction is stopped. Here you can see the three graphs showing the three factors affecting the rate of enzyme action. The first graph is of the temperature, the second one is of pH, and the third one is of the substrate concentration. Now the next topic, it's also very interesting and important topic from exams point of view, that is the mechanism of enzyme action. How enzymes they work, at what is the mechanism of enzyme reaction. Mechanism of enzyme action, we have discussed it in the first lecture also. Here you can see a diagram in which there is an enzyme and there is a substrate. There are substrate molecules. These enzymes and substrate molecules they will combine with the enzyme and a complex is formed that is called enzyme substrate complex. These substrates then they will change into products and the enzyme it will released. So this is the mechanism of enzyme action, how the enzymes they works. There are two models for the mechanism of enzyme action. The first model is called lock and key model and the second model is called induced fit model. First of all, we will discuss the lock and key model. This model was first given by a German chemist Emil Fischer in 1894, and according to this model, both the enzyme and substrate they possess specific shapes that fit exactly into one another. This model explains the enzyme specificity. Here you can see a picture. You can see there are two substrates, 1 and 2, and there is an enzyme. The substrates they have specific shape. Similarly, the enzyme's active site it also has a specific shape. So when the substrate molecules they will combine with the active site, there is a complex is formed that is called enzyme substrate complex. The reaction goes on and these reactants they are converted into products and the enzymes it will become separate. According to Emil Fischer, enzyme it work as a lock and substrate it work as a key. Similarly, every lock it has a specific key. The key has a specific shape according to its lock. When we enter that specific key to the lock, then the lock will open. Similarly, when the specific substrates, they will bind to the specific enzymes or enzymes having the specific active sites, then the enzyme will perform its function. Otherwise, there will be no rate of reaction. This model shows both the substrate and the active site of the enzyme. They have specific shapes. There is no change in the shape of both of these two molecules. And this model also explains enzyme specificity. It means every enzyme, it works, it attach with a specific substrate. This is the second figure showing the lock and key model. Here you can see the key act as a substrate molecules, they act as a key, and the lock means enzyme molecules, they act as a lock. Whenever a specific specific shaped substrate, it will enters the specific shaped enzyme, then the lock will open and the reaction will goes on. But if there is a change, there is a variation in the shape of both the substrate and the lock, then the key will not open the lock and there will be no process. Now the second model, induced fit model. This model was given by an American biologist, Daniel Hochland. Suggested a modification to lock and key model, and he proposed induced fit model. According to this model, active site is not a rigid structure. Rather, the active site of an enzyme, it molded itself according to the required shape of the substrate and then perform its function. Induced fit model is more applicable or more acceptable than lock and key model of enzyme action. Here you can see a yellow color enzyme. The enzyme has three specific points A, B and C. And you can see that the substrate has a slightly different shape. According to the lock and key model, this substrate will not fit into the enzyme. But according to a induced fit model, the substrate can fit into the enzyme. How? The enzyme's active site will arrange itself according to the substrate, and then the enzyme substrate complex is formed. And in this way, the rate of reaction, it will goes on. Hope you all will understand these important topics. Now the lecture is over. Thank you very much. If you have any queries, you can ask questions from me. Now there is a home assignment for you. From your exercise, there are the understanding the concepts. And under the heading of understanding the concepts, there are different questions. You have to attempt the question number 1, 2, 3 and 4 on your notebooks or loose sheets or your registers, whatever you have. And the second one is the short questions. There are 2 short questions in your exercise. Question number 1 and question number 2. You will attempt the question number 1 and question number 2 of short questions on your notebooks or loose sheets. That's all. In the next lecture will be the last lecture of this chapter, and at the end of the chapter I'll give you the important questions from the FBISC past papers, and all of you must solve those questions on your neat notebooks. Hope you all you guys will understand these topics very easily. If you have any problem, you can ask me or you can message me. Thank you so much. Allah Hafiz.